So today I'm going to be talking about a new probe that we're developing with the intention of clinical in vivo use. So as the title of this talk suggests, this probe measures scattering. When you measure scattering, what are you usually interested in? The first thing that probably comes to your mind is optical properties, like mu(s) prime or g. These are very important, and for several decades, numerous studies have shown that there can be great markers of many diseases and biological processes in all different types of tissue. But with this probe, I'm interested in going a little bit deeper than mu(s) prime or optical properties. And as an example of what I mean, I'm going to use these two samples, these two samples to give an example. So these are two polystyrene microsphere solutions, and I want to build a probe that can tell the difference between these. So when you look at them, they look identical. And if you were to measure their optical properties at a particular wavelength, they have identical optical properties. But they're different. The sphere sizes of each of them are actually very different. And they are not just structurally different, they are optically different as well. So if you look at their phase functions, they have very different phase functions. Although the general shape is the same, which is why they have the same optical properties, they obviously have very different fine structures. Now, if we're talking about building a probe for in vivo use, measuring the phase function with high resolution is very, very difficult, if not impossible. So typically what you measure with diffuse reflectance, you measure multiple scattering, not single scattering, which a phase function is a reflection of single scattering. So the question is, can you tell the difference between these two samples from multiple scattering? So we can scale this up with Monte Carlo from single scattering to multiple scattering and look at the impulse response of these two samples and look at the rotationally averaged impulse response, or P of R, or diffuse reflectance profile. So as you can see, this profile shows that they are different, and you can absolutely detect the difference between these two samples from multiple scattering. So that's kind of the motivation of this talk, is measuring or sensing the phase function from measurements of multiple scattering or diffuse reflectance. I'm going to take a step back and just talk briefly about the origin of light scattering in tissue and why that's important, and then I'm going to introduce you to our probe and give some results from experimental tests and simulations. And finally, I'm going to talk about our intended application for this probe. So when we think about scattering in tissue, where does it start? Well, it starts with the structures that actually scatter light. So if we take the structural properties of tissue and we can, let's say, take the mass density distribution and take the autocorrelation function of that, represented here by Bn of R, this is analytically linked to the phase function through the first Born approximation or the weak scattering approximation. It's a very simple process. It's just a Fourier transform. Then through numerical methods like Monte Carlo, we can get multiple scattering or the impulse response or spatial reflectance profile. So this implies that if you measure multiple scattering, you have sensitivity to single scattering or the phase function, which is analytically linked to the actual structures scattering light. So as an example here, I show on the left three different numerically generated media representing three different tissue types. And then using Born approximation, we can calculate the phase function from these three tissue types. That's shown in the middle. And then on the right is the rotationally averaged impulse response, or P of R. So the question is, if you want to measure this P of R and have sensitivity to these different, be able to distinguish these different tissue types, how do you measure P of R? So it's fairly obvious. You have to measure in the sub-diffuse regime. This means less than 1 mean free path from the illumination source, typically maybe 0.5 millimeter to 1 millimeter in tissue. So we designed a very simple probe to do just that, a very simple design. It's just 4 linearly aligned fibers. These fibers are 50 micron core with 5 micron cladding on each side, so it's like 60 to 65 micron center to center spacing. We illuminate the first fiber and we collect the three from the 3 subsequent fibers. So from one end to the other end, it's about 1/4 of a millimeter. So This is what data from this probe can actually look like. This is actually a measurement of tissue inside my cheek. So you can see there's the characteristic hemoglobin dips and the scattering spectrum in the red and near infrared. So as you can see, as you move further away from illumination, unsurprisingly, your scattering spectrum decreases, and so does your absorption spectrum. So the first characterization experiment I did with this probe is just to measure a dilution of microspheres, so just microsphere solutions with different μs prime. The μs prime spectra of these solutions is shown in the top right, and then on the bottom is data from the actual probe, each subplot representing a different fiber. So unsurprisingly, the intensity scales linearly with μs prime. But as I said in the beginning of this talk, I'm interested in more than just μs prime. I'm interested in going into the phase function. So going back to this example, can the probe distinguish these two microsphere solutions? So on the right, I show the phase function, which I showed earlier, and then on the left is data from the probe. So you can see that it can very easily distinguish these two solutions, and they have different shapes as you go further away from the source detector, as you increase source detector separation. And as you get closer to the diffuse regime, you can see that the two curves converge because and at that point, you're really measuring just high order scattering, which is losing its sensitivity to the phase function. So to get a little bit of a deeper investigation of what this probe can actually measure, I use Monte Carlo to actually model the geometry of the probe, meaning the fiber size and the NA of the fibers. So on the right, I show experimental results and simulation results of 3 phantoms that I measured and then used simulation to compare to. And as you can see, there's a very reasonable match between simulation and experiment. So thinking about in vivo applications, one question is that's important is depth sensitivity. So as you move further from your illumination source and you collect light, your light on average is going to come from deeper in tissue, which is shown here. This is important for tissue because if you're interested in a particular process, most likely it's not distributed homogeneously, infinitely in depth. It's at some localized depth. So I'm interested in my probe. What is my probe measuring? And then also, so how is that really what I want for my application? So this shows simulation from, on the x-axis is the max depth of photons collected by each fiber. And then on the y-axis is a normalized photon count. So as you can see, the further fibers have a very broad distribution, with the very close fibers having very narrow distribution. So with this kind of information, you can customize the probe to whatever depths you're interested in. But really the main question I want to answer with simulation is actually, like I said, what structures am I sensitive to, or particularly what size of structures? So I'm interested in the length scale sensitivity. So how I approach this question is with a perturbation study. So on the left is just some numerically generated media representing tissue. And then I pass that media through a low-pass filter to remove small structures. And then I pass it through a high-pass filter to remove large structures. And my goal is to see if the probe can actually detect these perturbations. So what does this mean optically? This means the spectral density function, or phase function, is being modulated by this exponential term. So on the top is the low-pass filter, and on the bottom is the high-pass filter. So this parameter W shown here is the characteristic size of the structures you're removing. So if I set W to, let's say, 500 nanometers, the low-pass filter removes all structures 500 nanometers and smaller. The high-pass filter removes all structures 500 nanometers and greater. So like I said, so then I can feed that into Monte Carlo and see if the probe can actually sense that. So this is the result. This is just an example of what this looks like in a phase function. So I think you're probably familiar. Small structures are responsible for a lot of backscattered intensity. So removing those structures kind of changes the backscattering end of the phase function. Removing large structures changes the forward scattering into the phase function. So now this is results from the Monte Carlo simulation. So the blue line here, what I show is 5% difference from not removing any structures. So I consider this what's experimentally measurable given noise and variability in a clinical system. So looking at the right figure, which is the upper length scale sensitivity, I remove 3, 5, and 10 micron structures. And as you can see, the 10 micron structures is just on the edge of what's experimentally measurable, meaning more than 5% difference. So I'll consider this the upper end of my length scale sensitivity. And then now let's look at the left. So this is what I'm really more interested in because of my clinical application, which I'm going to talk about in the next few slides, but the lower length scale sensitivity. So I remove structures 100 nanometers, 50 nanometers, and 25 nanometers. And all of these are detectable with 25 nanometers being just on the edge of sensitivity. I show 25 nanometers here just because it's a nice round number, but I found it's actually just about 24 nanometers. So the sensitivity is about 25 nanometers to up to several microns. So what is the application of this probe? So my group, we're interested in early cancer detection, or more specifically, detecting field carcinogenesis. So field carcinogenesis is this idea in cancer that in an affected organ at a very early time point in cancer progression, there are these very small structural changes, and actually subdiffractional structural changes throughout that organ. So for example, in colon cancer, which is something my group works with a lot, the early stages of the cancer start with precancerous lesions localized somewhere in the colon. These are called adenomas. So while these adenomas are localized to one point throughout the colon and rectum, there are these very, very small structural changes in the tissue that are subdiffractional, and they're not apparent in traditional histology. These changes are range from 10 nanometers to 100 nanometers in size, so within the sensitivity of this probe. Specifically, these changes are changes in size and shape of organelles, changes in the organization of microvasculature, and changes to the organization of the extracellular matrix. So, we did an ex vivo study to test the sensitivity of sub-diffuse reflectance to these changes. So we have an instrument in our lab, a technique we use called EBS, which measures P of R, or spatial reflectance in Fourier space. So in a biopsy study for pancreatic cancer and colorectal precancerous lesions, we find that P of R is substantially changed. And what's important to note here is that it's changed at very, very small length scales. So in the middle figure, this is the subtraction from the control P of R and the cancerous or precancerous P of R. So as you can see, the biggest difference is at very small length scales, indicating that the changes are a result of small structures and mostly changes to the phase function, not changes to US prime. So this is what the probe is optimized to detect. Now, in conclusion, I want to say that this probe obviously is a very simple design, but that's really our goal, is to make something that's really targeted at what we want and very powerful. It's very powerful in what we want to do, which is detect these very small structures. And as I've shown, its sensitivity ranges to about an order of magnitude lower and higher than the wavelength of light. So in the end, I want to say thank you for listening to my talk, and also thank you to everyone in my lab who contributed to this project, and I'd be happy to take any questions you have. Thank you.